and then making a vaccine based on spike protein and some of the hurdles we have in this vaccine production line. Okay, so this slide is really just about sort of reflecting on how amazing vaccines are and how lucky we are to have them. They've eradicated some diseases and dramatically reduced others, and those as well that are associated with cancer, such as HIV. They've increased our life expectancy by approximately 35 years, and they've improved our quality of life and suffering. I think this is a dramatic example of what's happening now, is what happens when we don't have a vaccine. And in the past, the world was rife with these diseases and still is in some countries. So I was just wondering if anyone could just name the first disease there on the left. Oh, I did click record and now it's stopped. Oh, I think it is recording. Yes, recording. Yes. And then the second one. Yeah, it is. So the first one is polio and the second one is tetanus. The third one?
Smallpox.
Yes, great. And the fourth one? Diphtheria. Yes, thank you. So diseases that we don't really see anymore. Why is vaccine production so long? You know, there's a lot of things in the news about it, and it's saying it normally takes 10 to 15 years, and we're trying to get this COVID-19 vaccine done in record time, but we still have a lot of diseases that we don't even have vaccines for. And just looking back through history, the vaccine development process has been standardized and regulated for good reason. And the diseases that we don't have vaccines for, HIV, RSV, malaria, just to name a few. So the earliest vaccine accounts go back as far as 200 BC and probably even before then. There are accounts of people creating their own smallpox inoculation in China and India, where they would use scabs from smallpox and ground this material up and then inhale it. And then in 1706, slaves in Boston were found to have scars from smallpox variolation in Liberia. So it's not really a new thing, but Edward Jenner in the was in 1796 or something like that. I've forgotten the actual date. Oh, I did go back. Let me just go back. The date is supposed to be on there, but I can't see it. Anyway, he was heralded as the founder of vaccines, as he created the first smallpox vaccine, which was then later used to eradicate the disease altogether. And he did this as he saw that there was a milder form of smallpox in cows, and milkmaids were getting these sort of boils and blisters on their hands, but it was nowhere near as bad as smallpox. And he took some of the pus from one of the milkmaids' blisters and rubbed it into the arm of an 8-year-old boy, James Phipps, and then he exposed him to the smallpox a few months later, and he didn't get ill. He reproduced this in several other cases and then published his work, which has, as I said, has led to the eradication of the disease by 1980. But there were still many issues at the time. Edward Jenner was treated like royalty. He was a doctor that lived in Cheltenham, but there were many issues, including funding, manufacturing, safety concerns, and deep-seated public fears, really, with the inoculation process. And this has really dominated vaccine throughout history. And then I just thought it was quite interesting as I read where the name vaccine came from. So variola, he called it the Variola vaccina, and variola is the Latin word for spot, and vacca meaning cow. So that's where our name vaccine comes from now. So that's quite interesting. I think Pasteur deserves a mention, as he was a French biologist, and he really made quite a few groundbreaking discoveries as he found he discovered the idea that germs are invisible to the naked eye, and the need for pasteurization of things like milk. He observed in 1897 that old bacterial cultures lost their virulence, and he created the chicken cholera vaccine out of this live attenuated sort of theory. And many of the vaccines that we use today are still carrying through this science. Can anyone name some of the live attenuated vaccines that we have today? I never really remember very well these. Yes.
MMR?
Yes. Good. So the TB1, BCG, MMR, polio. Yes. Good. Varicella. Right, next one. At the end of the 19th century, we had several vaccines for humans being developed and listed there, but these were biological agents, so they're quite dangerous. Unlike drugs, which are chemical agents, vaccines use live bacteria, viruses, toxins, so you can see the potential for things to go wrong in the production line. And they also use animals often, and there's a really fine balance with vaccines between producing immunity and not producing the disease and not causing a dysregulated immune reaction. The UK eventually set up the Division of Biologic Standards Acts in 1898. America was a bit behind and they had a few disasters. There was a horse, a milk wagon horse, who was then used to create gallons and gallons of diphtheria antitoxin for the antitoxin injection. But in 1901, he showed signs that he'd contracted tetanus and was euthanized. But it was too late, unfortunately, because this contaminated vaccine was distributed and 13 children in St. Louis in the USA sadly died from tetanus. And a similar incident happened in Camden in the USA where 9 children died from tetanus. So it's really quite a tragic history, and the Biologics Control Act came about as a result of that, where the government was able to regulate the sales of viruses, toxins and analogous products and control the vaccine production lines much more closely. But unfortunately, there was another incident, and again, this was in the US. There were several other incidents, but I'm just talking about some of the really big ones. In 1955, the USA was making the inactivated polio vaccine, and this was in the Qatar laboratory. Unfortunately, what happened was 200 children actually developed real polio because the vaccine was improperly inactivated, and they all had varying degrees of paralysis, and 10 died. So that was a real tragic event, and the vaccine was then replaced with an attenuated strain, but that strain that was given orally did sometimes become activated as it passed through the gut and still led to cases of polio in the 80s and 90s. But the Qatar legacy did lead to improved government legislation of vaccines, and it's made it one of the safest, most legislated processes in the medical industry today. It came, the Centre for Biologics Evaluation and Research, which is now the FDA, came about, and in the EU we have the European Medicines Agencies regulating vaccines, and the World Health Organization makes recommendations for biologic standards that many countries adhere to. But some of the negative things, other than the fact that the children got polio, is that there was a ruling that the Qatar laboratory had to pay compensation to the families of the victims, even though when they investigated them, they weren't found to be negligent. It was an accident that they couldn't have avoided, apparently. So, but they actually did have to pay a lot of money, which put a lot of companies off vaccine production because it's a low risk, low profit, high risk industry. So, and this has led to a lot of the shortages we see in vaccine production today. Only certain laboratories will make them, and the vaccine injury compensation program was made to try and help the manufacturing process so that they wouldn't be put off by this. But it still hasn't quite. There's still fear with companies. And then I think I couldn't talk about vaccines without talking about the MMR scare. I mean, we've all heard this many times, so I don't really need to go over it. But I think it's just a real example of public fear of vaccines and the delicate relationship they have with them, and parents being very protective of their children, who are the ones often exposed to these vaccines, even though they are amazing. So the vaccine development process is very long, as I've just said. It takes 12 to 15 years normally, and it starts with research in labs, then you go to preclinical trials where they test on animals and cell cultures, and then they start with small human trials to assess really the safety and the immunogenicity, and that is usually open label, and then they go on to larger trials and then even larger trials. And in the middle of this, they have to apply for the drug application, and then afterwards they have to apply for the license. The license can be difficult to get even after they've done all the groundwork. And the FDA then has the right to still inspect the factories where the vaccines are made and continue to do spot checks and even conduct their own testing on the vaccines. And then we have the vaccine's adverse events reporting system. About 30,000 events are reported a year, with 10 to 15 resulting in quite serious medical adverse events. And this is voluntary, anyone can report. And just an example of what happened with the rotavirus vaccine, in 1999 it was withdrawn because it was found to be associated with intussusception, and this was in 15 infants in the USA. So all of that's pretty exhausting and time consuming, and I take my hat off to anyone that's working on these vaccine trials, really, because they sound like you put in a lot of work, but you don't get much until right at the end. So just before I go on to talk about coronaviruses, I wanted just to talk about the different kinds of vaccines there are, quite important to cover. So we've got the whole organisms, which we had with the smallpox. Then we've got the toxoid vaccines, live attenuated, inactivated, subunit, and then we have the subunit and conjugate vaccines, and then we have reverse vaccinology. And these are really a reflection of the developing technology and science with time. So, for example, when they found out about the subunits, they found out that the immune system of the human immune system was making antibodies against the capsule of the bacteria, which was really great and allowed them to make those. But then they realized that when they put the capsule, when they injected the capsule, the immune system wasn't making a complete immune response to it, as it wasn't interacting with the T cells. So they then managed to add in a conjugate protein that allowed this antigen to react with the T cells and create a proper immune response. And then with reverse vaccinology, this really came about as we had advances in genomics, and scientists were able to really look at the whole genome of pathogens and study them in that way. And then we have the new vaccines that are coming out now, including the DNA vaccines, where pieces of genetic material to encode specific epitopes and antigens are directly injected into people's muscles, which sounds like crazy to me, but it is working according to their trials, and this is how they're trying to develop the HIV vaccine and also the COVID-19 vaccine. This is one of the techniques, and then there are vectored vaccines where they use a virus that's non-virulent or non-replicating to carry the bug into the cell. So yeah, in conclusion to vaccine development, they are tested in a very similar way to many other drugs, but even more so because they are biological agents, and they are given to large numbers of people, and so yeah, there has always been public opposition and fear. So yeah. Now I'm going to move on to coronaviruses. I'm not sure how much all of you know about coronaviruses. I was quite intrigued to find out why they're so successful and that kind of thing. So basically, they're enveloped viruses with a positive sense single-stranded RNA genome. There are 4 subgroups, alpha, beta, gamma, and delta, and these are divided according to their genetic similarity. And in humans, we are infected with the alpha and beta type. They primarily do cause infections in birds and non-human mammals, but since the 1960s, we've got now 6 strains that are known to cause infections in humans, and they're maintained in nature, which allows for genetic recombination and the emergence of novel viruses. I thought it would be interesting to just think about what kind of coronaviruses we were seeing before. These 4 strains were identified to be known to cause the common cold, but actually 2 of the strains do cause more severe disease in some children, those generally with underlying health conditions and immunocompromised and small babies. And I've definitely seen HKU1 coming up on some of the NPA results in some of the children I've had in the hospital, but it's never really been a really bad condition that they have had from it. And then we have the evolving coronaviruses, which are the more serious ones. And we have the SARS arising from China in 2003. And I've just put the number of deaths there, which seems like a really small number compared to what we're seeing now. And MERS that originated in Saudi Arabia. And these 2 strains of the coronavirus, which seem to sort of skip the species and jump to humans quite quickly, gave a clear warning of the lethality of certain strains. These were contained under high quarantine levels, but they left us knowing little about them and with no treatment or vaccines. And as we know, in late December, patients presenting with respiratory symptoms and fever due to an unidentified infection were found in Wuhan, which is the capital city of the Hubei province in China. And genome sequencing revealed the novel beta coronavirus strain in all 5 patients that they tested. And the WHO announced this as a public health emergency at the end of January. They called the disease COVID-19, and the virus classification system called it SARS-CoV-2. And this is similar to typical coronaviruses, where 2/3 of the RNA are translated into the proteins necessary for viral replication, and the 1/3 codes the structural proteins. And this is just a graphical representation of what it looks like, and the structural proteins are labeled there on the side. Now, my presentation from now on is going to focus on the spike protein. So this is the protein that protrudes from the membrane of the virus and gives it the appearance of a crown, and why it's called the coronavirus. It's really been used in a lot of the candidate vaccines because it has immunogenicity that's been proven, and it is the first contact that our cells have, so it allows viral entry into our cells. So if we can really stop it at this stage, it's felt to be a powerful potential vaccine. Now, the life cycle of COVID-19, how is it replicating and living in our cells? So it enters the human body, and the spike protein is cleaved by a human enzyme, which is thought to be this furin-like enzyme, but there are some other theories about which enzyme it is. This opens the fusion site, the receptor binding site of the spike protein, and allows it to bind to ACE2 receptors. It enters the cell by endocytosis, and the RNA genome is then released into the cytoplasm. And it uses our host ribosomes to make its transcriptase complex. And then it further hijacks our cell's machinery, rotating through the endoplasmic reticulum and the Golgi, making complete copies of itself. And finally, the viral progeny are transported by the Golgi vesicles to the cell membrane, where it is released into the extracellular space, ready to infect neighboring cells. And this is that picture again, showing the drugs that can work at different stages of the life cycle, and some drugs that can work on our dysregulated immune response. And these drugs are in trials now, and I think many of you will have heard in the news about remdesivir. I don't know how to say it, but it's actually shown to reduce the length of illness. This was made for the previous SARS and MERS and Ebola virus, but didn't have much success in them. But in the trials that they've done, it's shown to reduce the length of the illness by a little bit, which has been shown to be significant. So that's just quite interesting. Now, just a couple of studies just to show the immunogenicity of the spike protein. So this is a small study that was done in 2 hospitals in Hong Kong. 16 patients had confirmed COVID-19 by PCR, and they tested the antibodies for the spike and the nucleocapsid protein 14 days after the symptoms, and 100% of the patients had the immunoglobulin IgG for the anti-spike receptor binding domain, and 94% for the anti-spike RBD IgM. So this shows that, and less so for the nucleocapsid protein that you can see above that. So that's good, even though it was a small study. And also just to note that it has been found that patients with more severe disease and elderly patients do tend to produce more antibodies, whereas the people that don't get so severely infected don't seem to be producing as many antibodies. So I think that would be a different talk. So the vaccine test in monkeys was also done. This was with the SARS coronavirus, not the current coronavirus that we have. And they used this viral vector where a virus carries in the spike protein, a different virus that's not harmful, carries in the spike protein. They gave 4 monkeys this vaccine, and 4 received a control. And they showed that all the 4 monkeys that received the vaccine produced neutralizing serum antibodies against the spike protein and had no viral shedding, whereas the control group had viral shedding lasting 5 to 8 days. And I just wanted to briefly mention the ACE2 receptor, because I'd always just thought of this as being involved in blood pressure regulation and cleavage of ACE2, and why is the virus targeting this enzyme? But studies have shown that this enzyme has a non-enzymatic portion, and it's this non-enzymatic portion that the spike binds to. So this, so it's been looked at in quite a lot, this ACE2, and whether its expression is linked to predilection for disease, and where it is expressed, and is this why we're seeing lung disease and gastrointestinal symptoms, and just an interesting study showed that in animal models where the ACE2, basically the ACE2 was shown to have a protective role in lung injury and severe respiratory distress syndrome. So whether, it's just a really interesting talking point, I think, and research point. So steps in experimenting with the spike protein. So we know that the coronaviruses have the spike protein, but in order to make a vaccine for this new coronavirus, they needed to find the unique spike protein. So the researchers in China published the genome of the virus, and they released this. And one of the really good things about this virus, one of the good things, well, I mean, all of the research is free to read. None of it is paid for. None of the journals are locked or anything, because it's just in best interest of the public that everyone can have access to the research. So researchers then found the specific genes that coded for the spike protein, and they isolated them and made them in a lab, and then they injected these into mammalian cells in a lab dish, and those cells then produced the spike proteins. Then they really needed to know what the spike protein looked like, and specifically what the fusion site looks like when it's sort of open. So they used this cryon electron microscopy technique where they freeze the virus just as it's about to attach to the ACE2 receptor. And this allowed them to see what it looks like mapping each of its atoms in space. And they then sent these coordinates to dozens of research groups around the world. And one of the theories as to why this virus is so successful and so much more virulent than its sister virus, this SARS, is that it binds to this receptor with a 10 to 20-fold higher affinity, and we know it's infecting and killing so many more people. And this is one of the theories as to why, because it's very successful in its binding and fusion. So I'm just going to talk about a couple of trials now. There are quite a few trials going on, and more in America. So if you want to find out about all of those, or you know already about them, that's great, but I'm just going to focus on two. So this is the Oxford trial, and they've called the vaccine this very long name. And they're using an adenovirus, which has been taken from chimpanzees. So they're using a viral vector to take the protein from the spike of the COVID-19 into the host. And they have found that this develops a strong immune response, and because it's not a replicating virus, the adenovirus is felt to be safe. They've tested this in many animal species, and so far have recruited over 1,000 people aged 18 to 55, and the control group are going to receive a meningitis vaccine. The Imperial and St George's are also working with Oxford on recruiting for this study. They later hope to expand to adults and 50 and those of 50 years and above, and school-age children, and if it's shown to be safe and effective, to larger studies. And they also they want to test this on health workers, so you may be asked to join a trial, so beware. Imperial are testing a self-amplifying RNA virus, which is a cutting-edge vaccine, which, as I've described before, DNA is actually inserted directly into the host and into a muscle, and this is been found to generate the spike protein. It's expected to trigger an immune response and create immunity against the COVID-2. But as I've said, there are many other types of vaccine trials going on for previous SARS and for COVID-19, which I've listed there. And so just to mention, a lot of the studies have been tested on animals, but animal testing has a lot of problems. Mice, for example, make antibodies but do not develop pneumonia or SARS with the virus. And hamsters develop lung changes on the SARS infection but do not appear to get sick. And ferrets and several other monkey species develop lung disease but not consistently. So it's really impossible to predict exactly what effect this is going to have on humans, as it's very different what happens in our bodies. There's also been some animal studies that have shown an immunopathologic reaction. someone's asked a question there, which I'll come to at the end. It's quite an interesting question. In the 1960s, the RSV vaccine also had this immunopathologic reaction, where children that were given the RSV vaccine and then got RSV led to an 80% hospitalization rate with a much worse disease, and 2 of those children died. And that's why we don't have an RSV vaccine, because they haven't been able to really find a successful one, and we're just using the monoclonal antibody. This also occurs in dengue, where there is a hyperimmune response on re-exposure to a different strain of the pathogen. And then a similar effect has been observed in animal experiments giving the SARS vaccine and inactivated viral vaccines. It was felt that using a vector format could eliminate this problem, but we'll really need to see what happens now after the trials, and be aware of these things. And the vaccines have been proposed to be safe in some of the trials, but these have always been for very short observation periods. And as we know, the side effects and reactions can happen a long way down the line. Approval can be accelerated for vaccines, such as the flu vaccine, which is a well-known assembly line, where only 1 or few modules need to be changed each year. But the SARS is a novel pathogen, and the types of vaccine they're making for it are also very new, haven't really been used before. So the viral vector vaccine and the DNA vaccine. There's also a lot of politics involved when there's a pandemic going on, especially. Market power of pharmaceutical companies does delay responses in epidemics. They want to get money back for the investment that they've made. This happened in the case of the Ebola vaccine. There were lots of issues due to drug patents. And there is no money for pharmaceutical companies to make vaccines for very poor countries. And after the outbreak of SARS and MERS, there was a known need to develop a vaccine, but no pharmaceutical company would really invest in a vaccine that would potentially never be used. And there are some organisations out there that are helping with these hurdles. So CEPI is a coalition for epidemic preparedness, and Bill Gates is heavily involved in this organisation, and it gets funding from the World Health Organization, the Wellcome Trust, and donations, private and public. And their main goal is to really get the research and development incentives for these evolving pandemics, and to provide the money for vaccine production, and allow equitable access across the whole world, not just to the poor countries, to the rich countries. And ZAPI is an organisation which has an awareness of the fact that infectious diseases are very closely linked with animals and humans, and the ability that they can cross over, and brings together experts from the animal medical world and the human medical world. And just trying to be on top of these emerging zoonotic infections. Another problem with vaccination is the production line. So in order to make these vaccines, you can imagine that they're going to need vast production centres where they're going to be ramping up the number of vaccines they can produce. But they won't really want to do this until they know the vaccine is successful. Otherwise, it's just too risky. But they kind of need to do it quickly. So organisations like CEPI are trying to help with some of this risk, but they have put out a 2 billion call to help them do so. Seems to have frozen up. Nearly at the end now. Another potential problem with vaccines is distribution. So we know, even with the annual flu epidemic, that we vaccinate certain high-risk groups, so health workers, elderly people, underlying conditions, just because for cost reasons and supply reasons. And this is the goal, with keeping the sickness and the death rates as low as possible. But in a pandemic, the whole world is really going to be competing for these vaccines, and there's always an inherent imbalance where the poor countries can't access the vaccines as easily. And this occurred during the 2009 H1N1 flu pandemic. And Gavi, the Vaccine Alliance, is an organisation that does help get funding for the poorer countries and gets the vaccines to them. Okay, so that's my presentation, and just to summarise, I think we're really still trying to figure the virus out. The vaccine development is a risky business and has many aspects to it. the vaccine could save many lives, especially if the virus becomes endemic or perennially circulating. But it is likely that this pandemic will peak and decline before the vaccine is available. And until then, our best hope is to really just slow the spread and continue our good work in the NHS. And just to say thank you to the scientists. I'm sure I don't know if any scientists out there are listening, but they're always sort of hidden in the labs and they're always doing an amazing job. So that's it. So I don't know if there's any, there were some questions during the talk. Is the vaccine reported in the news where the first healthy volunteer died shortly after injection and a handful of other recipients had severe reactions? Oh, I didn't actually hear about that. That sounds really bad. Does anyone?
Laura, if you look at the link that I've put below,
yeah,
it was a fake news report.
Oh, goodness me. Okay. Well, that's good. What a relief. That's terrible fake news. Okay. Does anyone else have any questions? No? Okay. Well, thank you all for listening. And yeah, I hope you found it interesting and that you learned something. It's definitely kept me busy. So, yeah.
Well done, Laura. That was really thorough. Thanks.
Thank you. Bye. Bye, everyone.
Bye.
Bye.